supporting your professor, and for all the other guests of the Innovation Café. If anyone is watching online, thank you for being here. We are a weekly networking event where we offer presentations on the subjects of design and or technology. We are here every week. Next week we've got Johan Gilles here talking about his super formula. Afterwards we're very thankful to have Roy van der Meel here, of course.
Thanks for having me.
Afterwards, we'll have the opportunity to have some drinks and maybe also a meal. Because of a private event, it won't be located in the restaurant itself. We'll just have drinks here, and we've made a private room for the dinner. So if anyone wants to stay for dinner, let me know.
It's outside, so bring your jacket.
Yeah. And we'll first enjoy the talk. If you have any questions, there's room for that as well. And thanks also for your patience and for sitting out the technical issues, but enjoy the evening.
Nice your work. Thanks Anouk for the introduction, and welcome everyone online and everyone here. So I want to talk a little bit today about what we can or cannot do yet with RNA therapeutics. And I've been here before, so if you are a loyal follower of the Eindhoven Innovation Café, 2 years ago, I gave a talk, and this was only online because we're in the middle of a lockdown, on RNA vaccines. And you see that, especially for this occasion, I bought a new flannel shirt. So very closely related to what I'm talking about today. And for those online or here that don't know me, maybe a little bit redundant for most people that are here, this is my background. So I have a PhD in nanomedicine, and nanomedicine is basically the application of nanotechnology in medicine. So you can think of diagnostics or drug delivery. And then I did a couple of postdocs, and the most important one is that from 2015 to 2019, so just before the pandemic, I spent a couple of years in Peter Collis's lab, and his lab and associated biotech startups are responsible for developing the lipid nanoparticle technology that is now being used for the mRNA vaccines and other types of drugs. So in 2019, I was recruited by Professor Willem Mulder, who not that long ago also gave a talk at the Innovation Café. He talked more about the infrastructure that he's building, our research groups, but also Biotrip, the incubator that's there to commercialize our findings from the lab. So we moved from Vancouver to the beautiful Eindhoven, and now I live here with my wife and our twins. They're now 2.5. Maybe they're watching, so they're freaking out. They're seeing themselves on television at the moment. Obviously, this is not the Dak van Brabant or anything. This is in Austria, where my wife is from. And believe it or not, although this looks like the summer, we took this picture during Christmas. There was no snow. Anyway, more importantly for the research, this is our research group. So there's Willem there in the middle. And you see some of the people, or if you look behind you, you pretty much see half of them are sitting here. But what's important about this picture is that the Mulder lab is a combination of two research groups that is being managed as one, where we have a lot of talented young people working here around the corner in Eindhoven, engineering new nanotechnologies. And we have a group at the Radboud University Medical Center in Nijmegen, where of course you have the ability for translational work, immunology, animal models, patient materials, primary cells. So that's a little bit the how and where we work and all the people that are involved in this. So what we're talking about today is a new type of drugs. And in order to understand what we're talking about, we have to go all the way to the beginning, to the central dogma of molecular biology, which is the flow of genetic information. And all of our genetic information is caught in DNA, and DNA is in the nucleus of our cells. very well protected, the vault of our cells. So how does that information, that genetic information, actually flow to something functional? Well, there's the mRNA or messenger RNA intermediary, and from there you get a polypeptide, which then goes into protein. And protein is really are the functional effectors in our cells, in our body, and it's also what most conventional drugs target. So if you think of, a tablet that you might take, or if you think of biologicals like monoclonal antibodies, they all act on modulating the function of proteins. It can be by blocking them or doing something else to them. We're getting into the age where instead of, targeting proteins, we can also go before that. And by targeting mRNA, we can do two things. We can either, inhibit mRNA production, or we can promote it. And what you then basically do is you inhibit or you promote protein production, or in other words, you inhibit or promote gene expression. And what is important to know about doing or that strategy is that you don't mess with your genetic information that's caught in the DNA. You're only targeting the intermediary, so it's a transient effect. This is important to know. So in the next 15 minutes or so, these are the things that we're talking about. First of all, a little bit of nanotechnology. And I'll explain to you why we need nanotechnology for RNA drugs. Then I'll quickly give some examples what is now the current state of the art. And then we also venture in the last bit into, what is beyond the state of the art or what is currently happening. So why do we need that lipid nanoparticle technology that is used for the mRNA vaccines, for example? Well, you have to imagine that if you take something like an aspirin or paracetamol, there's an active substance in there, a small molecule that acts on these proteins that I was talking about before. And those molecules, when you take a pill and it falls apart in your stomach, it's taken up in your intestine, that molecule will then act or be able to act on the protein that it's needs to target for its therapeutic effect. And those small molecules are designed in such a way that they can also get into the cell. If we're talking about RNA, completely different beast. Much, much larger macromolecule. There is a heavy negatively charge on it, and which means that these RNA molecules cannot go into cells without help. And as you saw from the scheme before, the RNA that it's trying to target is in the cell. So you need a delivery system to protect that cargo and to get it in the right place. And that's what I'm doing or what I'm specialized at. So the nanotechnology that is most furthest evolved is the LNP platform technology, and where LNP stands for lipid nanoparticle technology. And this is a very wild, colorful scheme of how such a particle looks like schematically, based on microscopy images. And you have to imagine that these particles are only 50 to 100 nanometers in diameter. Let's say an order of magnitude smaller than what cells are, which makes sense because they have to be taken up by cells. So let's say 1/10 the diameter of a hair. And they contain different components. And some of these components may sound familiar. Cholesterol, it's naturally occurring in a body, gives these particles stability. There are phospholipids, and phospholipids are the components that make up the cell membrane. So that's all naturally occurring. Then there's this polyethylene glycol conjugated lipid that forms, let's say, a hydrophilic corona around these particles that basically prevents these particles from aggregating when you produce them. And importantly, there's an ionizable cationic lipid in there. don't have to go into the reason why that is there, but what's important, because the RNA is negatively charged, you need something that is positively charged to complex the RNA before the rest of the particle gets together. Now, these particles were developed to treat genetic diseases in the liver, or to deliver nucleic acid therapeutics in the liver. Why is that? If you look at this picture in the bottom here, that's let's say a microscopy image of the endothelium of the liver. So the lining of your blood vessels in the liver is fenestrated, which is another word for it has holes, right? And these holes are of a certain diameter because the liver is a filter organ, right? It filters stuff out of your blood. So if you inject nanoparticles in your bloodstream of a certain size, this size, and they pass the liver, they will get filtered out there and taken up by cells. So by taking advantage of the physiology of the liver and these materials, you can really deliver these big macromolecules to cells in the liver. So that's why we need nanotechnology. Protect the cargo and make sure that it gets delivered intracellularly. Now, what's the current state of the art? Mostly, as I said, delivering nucleic acids to the liver. And to give you an example of, inhibiting the production of mRNA or destruction of mRNA, so inhibiting protein production, is this drug called Onpattro. This came out at the end of 2018. It was developed by a company called Alnylam, based on this lipid nanoparticle technology that was developed in Peter Collis's lab. And when this came out at the end of 2018, I was there, and it was a huge success for the nanomedicine field and the nucleic acids therapeutics field, because it was the first drug that was able to deliver this type of nucleic acid therapeutic to the liver. And what it does is it is a lipid nanoparticle containing a small interfering RNA, and it's a tiny bit of RNA that targets a gene called Transthyretin, or TTR. That's maybe not something that you want to remember, but what you should remember is that for patients that suffer from this disease, or ATTR-mediated amyloidosis, there's a mutant protein production, which means that the protein doesn't fold well and causes plaque formation throughout the body. So if you think of Alzheimer's, where plaques form in the brain, in this disease, for these patients, these plaques or fibrils form all over the body, which leads to all sorts of problems: cardiotoxicity, polyneuropathy, you name it. It's an orphan disease, but it's a debilitating disease for people that suffer from it. And what is important here, there was no curative treatment for these patients before this drug came out. There are stabilizers for these proteins, but most patients that suffer from this, they need a liver transplant, and there's not that many livers to go around. So these are the results of a phase 3 trial on the basis of which this drug was approved. So this doesn't matter so much what this score is, but it basically says that if you compare the blue line and you see the treatment over time in this trial, so over 18 months, and these patients get a shot every 6 weeks of this drug, and in the gray line, this is the placebo group. And the only thing that you need to know from this scale bar is the higher the score, the worse the disease is getting. So you see that the patients that were getting this treatment was really stabilizing the disease, and in some patients actually caused an improvement. So that was the first example of an RNA drug that made it into the clinic, and that's only 4 or 5 years ago. There's also examples of promoting the production of mRNA. Anyone have an idea? I think so. Most of us got this jab of the mRNA vaccine. And I talked about this 2 years ago, how that works. Basically, we have now 2 mRNA vaccines for COVID that were developed in a very rapid fashion in less than a year. And that was, of course, standing on the shoulder of giants, 30 years of work into creating those nanoparticles for Onpattro. So it wasn't as easy as just swapping out that RNA drug that is causing a reduction in gene expression and just putting mRNA and that encodes for the spike protein. There was some re-engineering there, but the principle is the same. The only difference is that these particles with the vaccine, of course, you had them all in your shoulder. So they work a little different to induce an immune response there, an immune-specific memory. But if you would take these particles and you would inject them in the bloodstream, they would all end up in the liver. So that's the state of the art. We have 3 lipid nanoparticle-based drugs, 1 to inhibit gene expression in the liver, and 2 vaccines that you take that are given as a intramuscular injection. So where do we go from here? And there are multiple ways where the field is going, but one of the most interesting things maybe is now the ability to deliver the components to perform gene editing. So for those of you less familiar with what gene editing actually is, we talked about targeting proteins, right, with conventional drugs, biologicals or small molecules. We talked about the ability to either express or deliver mRNA that expresses for a protein, or to lower mRNA levels as a transient effect to inhibit protein production. But with gene editing, you can, in a very precise way, alter the DNA in our cells. So this means that we're going into the vault. We're really going into where your precious genetic information is. So it needs to be very specific because the changes that you make are permanent. And this is very important to realize, that the difference between gene editing that occurs on a DNA level and the, let's say, the RNA targeted drugs, there's a permanent versus transient effect. And that can have advantages and disadvantages. So, because nature videos can explain this a lot better than I do, I have a little video of how this works. Out. And when we're talking about lowering RNA levels, mRNA levels, we're talking about knockdown because it's transient, but here we're talking about knocking something out, something that's permanent. What you also heard in this talk, that you need to have two components there. You need to have the protein that is actually the scissors that makes this cut in the DNA, and you need a piece of RNA that guides this protein where to cut in the DNA. So that's already a lot more challenging than delivering just a piece of RNA. The second thing is, to reach the DNA, all of this needs to happen in the nucleus, which is a formidable extra challenge to get those two components in the nucleus to do this. But this is actually happening. So there's a company called Intellia, and they've taken this lipid nanoparticle that I talked about before, and they put these components in. So they've put an mRNA in, which encodes for this Cas9, so that's the scissors protein, let's say, and a guide RNA against the TTR gene, and that's what we talked about before with Onpetro. And then in the same way, these get infused, these particles then end up in the liver. When they get taken up by liver cells, they release the guide RNA, which is a small piece of RNA and able to go into the nucleus and also express those Cas9 proteins, the scissors, that are also able to go into the nucleus, cutting a specific piece of DNA. And because there are mistakes when the cell is trying to fix this, you knock this out. So why is this beneficial? I'll show you on the next slide. These are some results from their ongoing clinical trial that were reported last year or 2 years ago already, where you see here is a dose of the RNA that they're giving these patients, and these are groups of 3 patients, so these are really early stages. But what you see here is a mean reduction in the percentage of the TTR protein levels that they can find back in the circulation. And you see that for that higher dose, it's nearly down to anywhere between 90 and 100%. Now, what's important here is that what I mentioned before for Onpetro, these patients need to go to the hospital every 6 weeks and get an infusion for 45 minutes, have to be co-dosed with some corticosteroids because of that infusion. And for this, patients get 1 injection. Because you induce permanent changes, and this trial is actually ongoing already for longer now, and there's been a few dozen patients that have been dosed, and they see remarkable, long-lasting effects here. So that's going to be interesting in the future. Which disease do you want to treat with something permanent? Which disease do you want to treat with something that's transient? Because this you cannot undo. And of course, you know, transient has advantages in terms of, do you see side effects? You can change the dosing. So, there's one more video, and a small one. Let's say CRISPR-Cas9, what I showed you before, there can still be off-target effects. Here, this adds another level of specificity. So there are diseases that are caused by one single mutation in your DNA. Using what we call base editing, that is now, in theory, you can fix that. That's why it's called gene editing or gene repair. And what you can also do, and here's an example, is that you use that base editing to actually induce a single mutation, making a gene completely inactive. And a really nice example of that came out, or two papers actually came out 2 years ago, where they delivered, again, an mRNA encoding for this adenosine base editor and a short guide RNA targeting this gene here, PCSK9. Why is PCSK9 important? PCSK9 is involved in the production of your blood cholesterol. And as most of you probably know, you have your good and your bad cholesterol. There's millions of people taking statins every day over the world to lower their bad cholesterol, or otherwise known as LDL. So there's this company called RNA Therapeutics, and they use a lipid nanoparticle technology from Acutis, which is a spin-off company from the Collis Lab, that have basically started doing this. So not in humans yet, but in non-human primates, or in monkeys. 4 of these monkeys that get treated with this base editor, let's say, they see when they take out the liver that there's about 60% editing. So of all the cells that are in the liver, they're able to reach 60% editing. So what does that translate into? Well, if they give 1 injection and they take blood from these monkeys, from these 4 here, compared to the 2 that are untreated, and they look at the levels of the PCSK9 protein in the blood, they see that it's completely reduced up to 200 days. And that translates into a lowering of your bad cholesterol for a long, long time. This is supported by a paper that came out last week, that there are people that have natural mutations in the PCSK9 gene that have a 50% less chance of developing cardiovascular disease. So you have to imagine that instead of people having to take statins every day, drugs every day, that may not work so well, that these patients in the future might be able to be treated with a single shot. So, what are the challenges at the moment in this field, and also the things that we work on in our research group? So, if you have questions, ask the people behind you. What we didn't talk about is that we talked mostly about intravenous administration of these nucleic acid therapeutics. There are, of course, also people that work on ex vivo approaches, where you take cells out of the patient, change those cells and put them back. It's very successful. But what you really would like to be able to do is, when you inject these particles, that they not only go to the liver, but you'll also be able to target other organs. For example, the lungs, to treat cystic fibrosis, genetic disease, or target the spleen or the bone marrow. The challenges here is repeated dosing, especially of nucleic acids, but also these LNPs can cause immunostimulatory effects, or they cause immunostimulatory effects, right? We have evolved for millions of years to recognize when we get nucleic acids in our blood, our body thinks something is wrong, there's a bacterial or viral infection, and all alarm bells and red flags go off. However, while working on those things, there are also very, very interesting opportunities here. For example, think about personalized anti-cancer vaccines. You know that cancer is caused by mutations in the DNA. If you're able to sequence which mutations are responsible not only for the cancer, but what separates a cancer cell from a healthy cell, you will be able to make a cancer vaccine and direct your immune system to get rid of those cells. And of course, what we talked about a little bit here, the last example is an example of a one and done treatment, where you get a single injection and you basically provide patients with a curative therapy. So lots of exciting things. Hopefully in 2 years I'll be here again giving another talk about what has happened in the last 2 years. But for now, if you cannot get enough of this and your Dutch is getting better and better, there's a University of the Netherlands video where I also talk a little bit about this. And of course, we work on this with an entire group of people. I already showed them, but just to finish with this and acknowledge the support that we get from our funders, I'm really happy to be working with this group of talented people This is definitely not something that we can work on, by yourself. So thanks a lot for listening. Also, people online, and I'm happy to discuss or answer any questions. Thank you. Now I'm expecting lots of tough questions from my own group, but yeah, please let me know if there's anything you want to ask me.
Maybe I want to start. I have a question about first thank you for a wonderful talk. Actually, there's a lot of insight in the work you're doing. There's a lot of exciting opportunities
there is.
Right? I think there's one side of me that has questions about the societal scare also around it.
Sure.
We've seen this around RNA treatments as well, right? Not just the anti-vaxxers, but everything in between. That's also what is actually happening here, right? So there's I think a big question of translating some of this really exciting and detailed technology, but that also goes into, like you said, the vault of our life form.
Sure.
Right? And the implications of it. So I wonder.
Yeah, there's two things here, right? A couple of years ago, a Chinese researcher, this is a very famous case, edited cells in embryos to basically knocking out a receptor that HIV uses to gain entry into cells. Using that as a reason, or he edited cells in embryos that then grew up to be babies and are now a certain age. He went to jail for that. Mm-hmm. What we are talking about here is, of course, editing the cells, yes, in the vault, but to fix disease in a person that, of course, will not be transferred to your offspring. So that's one major difference. But you are right. That's why I mentioned the effects being from transient to being permanent. And it really, really depends on, I think, the type of disease where you say, okay, this is where we can make a permanent change, and it's possible that there are side effects, but disease itself is so serious or debilitating that one outweighs the other, right? The personalized cancer vaccines are a very good example of that. The reason why is when people nowadays talk about personalized treatment, you say, look, 2 patients with breast cancer come in, and one of them has a certain mutation, they get a certain antibody treatment, and maybe the other person doesn't have that. That's already a level of differentiating between those patients. But most people, they just get bombed with chemotherapeutics. And I think everyone knows how bad the side effects of chemotherapy can be, or are, because it goes everywhere. So imagine that you now go to a doctor, they take a biopsy, exactly the same as what happens now, but based on that biopsy, they develop a vaccine that is personalized, specifically geared towards your cancer. Yeah, I would definitely opt for that one rather than having my body being full of chemotherapeutics. That so many people were dosed with these lipid nanoparticles and mRNA as a vaccine due to COVID has also, of course, yielded a tremendous amount of information about what works well, but also what are side effects, not only of the mRNA that can be immunostimulatory, but also the components that I showed you in the LNP itself, right? For example, that lipid that is needed to complex the RNA, it has a positive charge. That doesn't really happen in nature. So that by itself will cause an effect in humans. And there, it's really about finding a balance between, dosing, therapeutic application, and I think that can heavily influence also the public opinion on using this technology. So I really, try to show what are the possibilities and what is going on. But it's not all about potency or therapeutic effects. The side effects, and especially on the immune system and also longer term, are something that's yeah, equally important, for sure.
Like what a wonderful kind of like side effect of this dramatic events, I guess, like around kind of like COVID, right? But to really
Yeah.
engage trust, like within really populations, like around, like.
It is, and it's good that people are critical because, you know, this was a completely new technology that was rushed in during COVID. And it's good that that discussion is there, and it also is good to, you know, to be critical about it so we improve on it, right? Evelina.
I have a question. So about your last slide with this potential application.
With these people?
No,
Okay.
For the anti-cancer vaccines, right?
Yeah.
Because it sounds very attractive, but isn't it that the limitation will be very similar as in the chemo, conventional chemo, because it will work only if every cell will get it, basically?
Yes and no, because what you do is not, you don't aim at the cancer cells, the actual vaccine. The vaccine is aimed at immune cells. And what you do is you try to switch on the immune system so it goes after specific cancer cells. The challenge more is that if you think of the immune response that you need to get from a vaccine versus something that you need to get in order to get rid of your cancer that may have spread throughout the body, I completely agree that those are two different challenges. And it's definitely not the case that we can just, you know, just take the COVID-19 vaccine as a template, you know, just switch out the mRNA and there's no more cancer in the world. Absolutely not. There is a lot of engineering needed, and it's also very much dependent on the type of cancer. We know that, right, from other immunotherapies, that some are very susceptible for these types of treatments, and others are just inaccessible to, or they're very good at, you know, fighting off the immune system. So it's not as simple as just making this particle and we cure cancer. There will likely be a combination therapy, maybe with chemo or other types of immunotherapies to do this. But the fact that it is much more targeted towards a patient's personalized mutations rather than just, you know, carpet bombing the body with chemo is, I think, a huge progress, in my humble opinion. More questions? And of course we can continue discussing over a drink, which I don't think is a bad idea.
Yeah, sounds like you're introducing. I was wondering, like, how long would it take to sort of introduce this vaccine for cancer?
Yeah, so I went to a conference last week, 2 weeks ago in Berlin, which was all about mRNA drugs. And some of the big companies were there, right? You know the names from the vaccines, the BioNTechs and the Modernas, and a term that they like to use at the moment is the 100-day challenge. The 100-day challenge means a patient coming into the doctor's office and getting a biopsy to having a vaccine ready. And that's, let's say, 3 months. And of course, for some cancers, that's fine. Other cancers, yeah, there might be people coming into the doctors that don't have 3 months anymore. But the 100-day challenge, and this is really going from, you know, not only taking the biopsy, but having to sequence that, identifying the mutations that are, you know, so a vaccine can distinguish, or your immune cells can distinguish between a healthy cell and a cancer cell, then producing it in a way that it's completely safe to use from the material side, and then of course having to treat the patient. So that's what they're currently aiming for, is the 100-day challenge. That would be very good. And also, so if you think about, or hopefully you don't think about that, but if you consider a next pandemic, or let's say the flu shot each year, the flu shot currently is, you know, they look at what they think is going to be the dominant strain of flu, they make a flu shot against it, or they combine a couple of strains, and that is about 50% effective. if you can make a flu shot in much less time, you can first look at one part of the world where the flu is coming up, then use an mRNA vaccine and maybe make that in a much shorter time and have a much, let's say, more potent vaccine that is maybe 70, 80, 90% effective. So it's not only about the cancer vaccines, but it's definitely also about vaccines for other infectious diseases, or hopefully not another pandemic. Let's put it that way.
Any more questions?
Well, just like you introduced just now, I think it's a good idea to mingle later on. Well, thanks for being here. For most of you, it was just a regular lecture, maybe.
Just a regular lecture from the
thanks
boss.
for being here. We are here every week, so if you like the concept and the idea of it, come again. We have a program up until the end of March. Take someone along with you. We're very happy to have you here. So I'm going to go and ask someone from the service to come and get some drinks. Later on, I will ask if you are interested in staying for dinner. Then, but just stick around and have a lovely evening. Thank you.
Okay. Thanks. Good. All right. Switch that off.
Well done, sir.
Now we know what I do.
Yes. No,